Hello everyone. Now we are going to start the new paper, paper 2 of life science. We'll be having a really quick recap of all the topics, very basic information shared because this is a quick recap session. So let's start with the first portion. Biology, molecules and interactions. Molecules and their interaction relevant to biology. Molecules and their interactions are essential for the functioning of biological systems. Here are some of the most important molecules and their interactions in biology, like proteins. Proteins are the most diverse and important molecules in biology. They are composed of amino acids and perform a variety of functions, including catalysis, structure, and signaling. Then nucleic acids. Nucleic acids, including DNA and RNA, are responsible for the storage and transfer of genetic information. They also play a role in protein synthesis and regulation. Then carbohydrates. Carbohydrates are important energy sources and structural components in many biological systems. They include sugars, starches, and cellulose. Lipids. Lipids are a diverse group of molecules that include fats, oils, waxes, and phospholipids. They play important roles in cell membrane structure, energy storage, and signaling. Then enzymes. Enzymes are proteins that catalyze biochemical reactions. They are important for many cellular processes, including metabolism and DNA replication. Then receptors. Receptors are proteins that bind to specific molecules such as hormones or neurotransmitters and transmit signals to the cell. They play a critical role in cellular communication and signaling. Then there are antibodies. Antibodies are proteins that are produced by the immune system in response to foreign molecules such as pathogens. Then hormones. Hormones are signaling molecules that regulate many biological processes, including growth, development, and metabolism. The interactions between these molecules are critical for many biological processes, including protein synthesis, cellular communication, and immune response. Understanding these interactions are essential for understanding the mechanisms underlying many diseases and developing effective treatments. Life science and its importance. Life science, also known as biology, is the study of living organisms, their structure, function, behavior, and interactions with the environment. It is an incredibly important field that impacts nearly every aspect of our lives, from medicine to agriculture to conservation. Here are some reasons why studying life science is important. Understanding life processes. By studying life science, you can learn about the processes that sustain life, such as cell division, metabolism, and DNA replication. This knowledge is essential for understanding how living organisms function and how they interact with the environment. Then, developing new medicines. Life science research has led to the development of many life-saving medications such as antibiotics and vaccines. By studying life science, you can contribute to the discovery of new drugs and therapies to treat a wide range of diseases. Now, improving agricultural practices. Life science research has also led to the development of new agricultural techniques and technologies such as genetically modified crops that can increase crop yields and improve food security. Then, protecting the environment. Understanding how living organisms interact with their environment is essential for protecting ecosystems and conserving biodiversity. Life science research can help us understand the impacts of human activities on the environment and develop strategies to mitigate them. Enhancing your critical thinking skills. Studying life science requires critical thinking, problem solving, and analytical skills. These skills are valuable in the wide range of professions, including medicine, research, and environmental management. Overall, studying life science is important for understanding the fundamental processes that sustain life and how we can use this knowledge to improve human health, agricultural practices, and environmental sustainability. Then, structure of atoms, molecules, and chemical bonds. Atoms are the basic building blocks of matter, and they are composed of three types of subatomic particles: protons, neutrons, and electrons. The protons and neutrons are located in the nucleus of the atom, while the electrons are found in the electron cloud. Protons and neutrons are located in the nucleus. We know that. And protons and neutrons are located in the nucleus of the atom, while the electrons are found in the electron cloud surrounding the nucleus. The number of protons in the nucleus determines the atomic number of the atom and the fourth identity of the element. For example, an atom with 6 protons is carbon, while an atom with 8 protons is oxygen. The number of neutrons can vary within an element, resulting in different isotopes of the same element. For example, carbon 12 and carbon 14 are isotopes of carbon with different numbers of neurons. The electrons are arranged in energy levels or shells around the nucleus, with the innermost shell containing 2 electrons and the outermost shell containing up to 8 electrons. The arrangement of electrons in the outermost shell, known as the valence electrons, is critical for chemical bonding. Molecules are formed when 2 or more atoms are held together by chemical bonds. There are 3 main types of chemical bonds, namely covalent bonds, ionic bonds, and hydrogen bonds. Covalent bonds are formed when atoms share electrons to achieve a full outer shell. For example, 2 hydrogen atoms can form a molecule of hydrogen gas by sharing electrons to form a covalent bond. The electrons are shared equally in a non-polar covalent bond, while they are shared unequally in a polar covalent bond. Ionic bonds are formed when 1 or more electrons are transferred from one atom to another, resulting in oppositely charged ions that are attracted to each other. For example, sodium and chlorine atoms can form an ionic bond to form a sodium chloride or table salt. Next, hydrogen bonds are weak bonds that form between a hydrogen atom with a partial positive charge and a nearby atom with a partial negative charge, such as oxygen or nitrogen. For example, hydrogen bonds are responsible for the unique properties of water, such as its high surface tension and boiling point. The structure of the molecules is determined by the type and arrangement of chemical bonds between atoms. For example, a molecule of water has 2 polar covalent bonds between the hydrogen and oxygen atoms, resulting in a bend shape and a partial negative charge on the oxygen atom and partial positive charge on the hydrogen atom. In summary, the structure of atoms, molecules, and chemical bonds is fundamental to the study of chemistry and the physical world around us. Understanding the arrangement and behavior of atoms and molecules is essential for understanding chemical reactions, material science, and many aspects of biological process. Then, structure of atoms, molecules, and chemical bonds. Atoms are composed of protons, neutrons, and electrons. The number of protons in the nucleus determines the atomic number and identity of the element. Electrons are arranged in energy levels or shells around the nucleus, with the outermost shell containing the valence electrons. Molecules are formed when 2 or more atoms are held together by chemical bonds. There are 3 main types of chemical bonds, like covalent bond, ionic bond, and hydrogen bonds. Covalent bonds are formed when atoms share electrons to achieve a full outer shell. Ionic bonds are formed when 1 or more electrons are transferred from one atom to another, resulting in oppositely charged ions that are attracted to each other. Hydrogen bonds are weak bonds that form between a hydrogen atom with a partial positive charge and a nearby atom with a partial negative charge. The structure of molecules is determined by the type of arrangement of chemical bonds between atoms. The arrangement and behavior of atoms and molecules are essential for understanding chemical reactions, material science, and many aspects of biological processes. Next, composition, structure, and function of biomolecules. So, biomolecules are the organic compounds that are essential to life processes. They can be classified into 5 main groups, like carbohydrates, lipids, proteins, nucleic acids, and vitamins. So, carbohydrates. Carbohydrates are biomolecules composed of carbon, hydrogen, and oxygen. They are classified into monosaccharides, simple sugars, disaccharides, 2 monosaccharides linked together, and polysaccharides, complex carbohydrates. Carbohydrates serve as a source of energy and play a structural role in cells. Examples of carbohydrates include glucose, fructose, lactose, sucrose, starch, and cellulose. Then lipids. Lipids are biomolecules composed of carbon, hydrogen, oxygen, but a very much higher proportion of carbon and hydrogen than oxygen. They include fats, oils, waxes, and steroids. Lipids serve as a source of energy, provide insulation, and form cell membranes. Examples of lipids include triglycerides, phospholipids, and cholesterol. Then proteins. Proteins are biomolecules composed of amino acids linked together by peptide bonds. They play a variety of roles in cells, including enzymes, hormones, transport, and structural proteins. Proteins are essential for growth, repair, and maintenance of tissues. Examples of proteins include insulin, hemoglobin, collagen, and keratin. Then nucleic acids. Nucleic acids are biomolecules composed of nucleotides. They are responsible for the storage and transmission of genetic information. There are 2 types of nucleic acids: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. DNA stores genetic information while RNA is involved in protein synthesis. Examples of nucleic acids include DNA, RNA, and ATP, adenosine triphosphate. Then vitamins. Vitamins are organic compounds that are essential for normal metabolic processes. They are classified into two categories like fat soluble and water soluble. Fat soluble vitamins include vitamins A, D, E, and K, while water soluble vitamins include vitamins B and C. Vitamins play various roles in the body, such as regulation of cell and tissue growth, maintenance of healthy skin, and prevention of infections. In summary, biomolecules play critical roles in the structure, function, and regulation of cells and organisms. Understanding the composition, structure, and functions of biomolecules is essential for the study of biochemistry, nutrition, and many aspects of human health and diseases. Next topic, stabilizing interactions, van der Waals, electrostatic, hydrogen bonding, and hydrophobic interactions, etc. Stabilizing interactions are the non-covalent forces that are used to hold biomolecules such as proteins and nucleic acids together in a specific structure. These interactions include van der Waals, electrostatic, hydrogen bonding, hydrophobic interaction, and others. So first, van der Waals forces. Van der Waals interactions are weak interactions or weak attractions between two molecules that arise from the fluctuation of electron distribution around atoms. They are the weakest of all the stabilizing interactions but can play a significant role in the stabilization of biomolecules. Van der Waals interactions are important in protein-protein, protein-DNA, and protein-ligand interactions. Then electrostatic forces. Electrostatic interactions, also known as ionic bonds, are the attraction between oppositely charged ions. They are relatively stronger and can play a significant role in the stabilization of biomolecules. Electrostatic interaction are important in protein-protein and protein-nucleic acid interactions. Examples of electrostatic interaction include the attraction between the positively charged amino acid arginine and the negatively charged amino acid glutamate. And then hydrogen bonding. Hydrogen bonding is the interaction between a hydrogen atom covalently bounded to a highly electronegative atom such as oxygen or nitrogen and another electronegative atom. Hydrogen bonding is relatively strong and plays a significant role in the stabilization of biomolecules such as the alpha helixes and beta sheets in proteins and the base pairing in nucleic acids. Then hydrophobic interactions. Hydrophobic interaction occurs when non-polar molecules cluster together in an aqueous environment to minimize their contact with water. Hydrophobic interactions play a significant role in the stabilization of proteins and other biomolecules. Hydrophobic interactions are important in protein folding and the formation of lipid bilayers. In summary, stabilizing interactions are crucial for the proper folding and functioning of biomolecules. Understanding these interactions is essential for understanding the structure and function of proteins, nucleic acids, and other biomolecules. Then principles of biophysical chemistry like pH, buffer, reaction kinetics, thermodynamics, colligative properties. So biophysical chemistry is the study of the physical and chemical principles underlying biological processes. Some of the key principles in biological chemistry include pH, buffer, reaction kinetics, thermodynamics, and colligative properties. So first, pH. pH is a measure of the acidity or basicity of a solution. It is defined as negative logarithm of the concentration of hydrogen ions in a solution. pH plays a crucial role in many biological processes including enzyme activity and protein folding. Maintaining the proper pH is essential for the proper functioning of cells and organisms. Then buffer. A buffer is a solution that can resist changes in pH. Buffers are essential for maintaining a stable pH in a biological system. Buffers work by absorbing or releasing hydrogen ions. Before moving on to the complexity, let's once again read buffer. A buffer is a solution that can resist changes in pH. Buffers are essential for maintaining a stable pH in a biological system. Buffers work by absorbing or releasing hydrogen atoms in response to changes in pH. Examples of buffers in biological systems include the bicarbonate buffer system in the blood and the phosphate buffer system in cells. Next topic is reaction kinetics. It is the study of the rate and the mechanism of chemical reactions. Understanding reaction kinetics is essential for understanding the behavior of biological molecules such as enzymes and proteins. Reaction kinetics can be described using various models such as the Michaelis-Menten model, which describes the relationship between substrate concentration and enzyme activity. Next, thermodynamics is the study of the energy and entropy of systems. Understanding thermodynamics is essential for understanding the stability and behavior of biological molecules. Thermodynamics can be used to predict whether a reaction will occur simultaneously or require energy input. The Gibbs free energy is a thermodynamic quantity that can be used to predict the direction and spontaneity of a chemical reaction. Then colligative properties. Colligative properties are properties of a solution that depend on the number of solute particles present rather than the identity of the solute. Examples of colligative properties include freezing point depression and osmotic pressure. Colligative properties play a critical role in many biological processes, such as the regulation of water balance in cells. In summary, the principles of biophysical chemistry are essential for understanding the behavior of biological processes and underlying physical and chemical processes that govern biological systems. These principles have important applications in fields like medicine, biotechnology, and bioengineering. Colligative property. Colligative properties are physical properties of a solution that depend on the concentration of solute particles in the solution rather than on the chemical identity of the solute itself. There are four main colligative properties. Vapor pressure lowering. Vapor pressure is the pressure exerted by the vapor phase of a liquid in equilibrium with the liquid phase. When a non-volatile solute, a solute that does not readily evaporate, is added to a solvent, the vapor pressure of the solvent is lowered. This is because the solute particles occupy. What's happening here? When a non-volatile solute, a solute that does not readily evaporate, is added to a solvent, the vapor pressure of the solvent is lowered. This is because the solute particles occupy some of the surface area that would otherwise be occupied by solvent particles. Vapor pressure is the pressure exerted by the vapor phase of a liquid in equilibrium with the liquid phase. When a non-volatile solute, a solute that does not readily evaporate, is added to a solvent, the vapor pressure of the solvent is lowered. This is because the solute particles occupy some of the surface area that would otherwise be occupied by solvent particles. As a result, fewer solvent particles are able to escape into the vapor phase and the vapor pressure is reduced. The degree of vapor pressure lowering depends on the concentration of solute particle in the solution. Then boiling point elevation. Next, colligative property is the boiling point elevation. Then boiling point elevation. The boiling point of a liquid is the temperature at which its vapor pressure equals the atmospheric pressure. When a non-volatile solute is added to a solvent, the boiling point of the solvent is raised. This is because the solute particles decrease the vapor pressure of the solvent, meaning that the temperature must be increased to achieve the same vapor pressure. The magnitude of the boiling point elevation depends on the concentration of solute particles in the solution. Next stage is the freezing point depression. The freezing point of a liquid is the temperature at which it transitions from a liquid to a solid. When a solute is added to a solvent, the freezing point of the solvent is lowered. This is because the solute particles disrupt the formation of crystal at the structure of the solvent, making it more difficult for the solvent particles to form a solid. As a result, the temperature must be lowered further to achieve the same transition from liquid to solid. The degree of freezing point depressions depend on the concentration of solute particle in the solution. Next, colligative property is the osmotic pressure. Osmotic pressure is the pressure that must be applied to a solution to prevent the net flow of solvent molecules across a semi-permeable membrane. When a solute is added to a solvent, water molecules tend to flow from the side of a membrane with low solute concentration to the side with higher solute concentration. This flow of water is driven by the tendency of the system to achieve equal solute concentration on both sides of the membrane. The osmotic pressure is proportional to the concentration of solute particles in the solution. In summary, colligative properties are physical properties of a solution that depend on the concentration of solute particles rather than on the chemical identity of the solution itself. These properties have important applications in fields such as chemistry, biology, and materials science. Next, we are going to learn some topics such as bioenergetics, glycolysis, oxidative phosphorylation, coupled reaction, group transfer, biological energy. Transducers. So, bioenergetics refers to the study of how living organisms acquire, transform, store, and use energy. It encompasses a wide range of processes including glycolysis, oxidative phosphorylation, coupled reactions, group transfer, and biological energy transduction. Glycolysis is the initial step of glucose metabolism, which involves in the cytoplasm of cells. It involves the breakdown of glucose into two molecules of pyruvate with the net production of two molecules of ATP and two molecules of NADH. Glycolysis is a universal pathway found in all living organisms, and it serves as a source of energy for many cellular processes. Next, oxidative phosphorylation. Oxidative phosphorylation is a process that occurs in the mitochondria of eukaryotic cells and involves the generation of ATP through the oxidation of NADH and FADH2. The electron transport chain, or ETC, is the primary mechanism for oxidative phosphorylation, where electrons are transferred through a series of protein complexes and ultimately used to pump protons across the mitochondrial inner membrane. This creates an electrochemical gradient that drives the synthesis of ATP by the ATP synthase enzyme. Oxidative phosphorylation is responsible for the majority of ATP produced by aerobic organisms and plays a critical role in cellular respiration. Then, coupled reactions. Coupled reactions are a set of chemical reactions where the energy released by one reaction is used to drive another reaction that requires energy input. This allows the energy from exergonic reactions to be harnessed to drive endergonic reactions. An example of the coupled reaction is the hydrolysis of ATP, which releases energy that is used to drive other cellular processes. Then next topic, group transfer. Group transfer reactions involve the transfer of functional groups from one molecule to another. For example, the transfer of phosphate group from ATP to another molecule is a common type of group transfer reaction that is used to transfer energy between molecules. Then next topic, biological energy transduction. Biological energy transduction refers to the process by which cells convert energy from one form to another. An example of biological energy transduction is the conversion of light energy into chemical energy during photosynthesis. In this process, energy from sunlight is used to drive the synthesis of ATP and NADPH, which are used to power other cellular processes. In summary, bioenergetics encompasses a range of processes that are essential for life, including glycolysis, oxidative phosphorylation, coupled reactions, group transfer, and biological energy transduction. These processes are critical for the acquisition, transformation, and use of energy by living organisms, and they have important applications in fields such as medicine, agriculture, and biotechnology. Next, principles of catalysis, enzymes and enzyme kinetics, enzyme regulation, mechanisms of enzyme catalysis, isozymes. Principles of catalysis, enzymes, and enzyme kinetics are the fundamental concepts of biochemistry that explain how chemical reactions occur in living organisms. Here, a detailed explanation of each of these topics. Principles of catalysis. Catalysis is the process of increasing the rate of a chemical reaction by lowering the activation energy required for the reaction to occur. Enzymes are biological catalysts that facilitate chemical reactions by reducing the activation energy required for the reaction to take place. This is achieved by stabilizing the transition state of the reaction and increasing the proximity and orientation of the reactants. Enzymes and enzyme kinetics. Enzymes are protein molecules that catalyze specific chemical reactions in the body. Enzyme kinetics is the study of the rate at which enzymes catalyze reactions, and it provides insight into the mechanism of enzyme catalysis. Enzyme kinetics involve measuring the initial rate of reaction as a function of substrate concentration and enzyme activity. Then enzyme regulation. Enzyme activity is regulated by a variety of factors, including the concentration of substrates and products, changes in pH or temperature, and the presence of regulatory molecules or cofactors. Enzyme regulation is essential for maintaining cellular homeostasis and controlling metabolic pathways. Then mechanism of enzyme catalysis. Enzyme catalysis involves a complex series of events that occur between the enzymes and the substrates. This includes substrate binding, catalytic transformation, and product release. Enzyme catalysis is often described using a lock and key model where the enzyme's active site is complementary in shape to the substrate. This allows the enzyme to bind to the substrate and facilitate the chemical reaction. Isozymes. Isozymes are enzymes that catalyze the same reaction but have different amino acid sequences and structures. Isozymes can have different catalytic properties, substrate specificities, and regulatory mechanisms. This allows cells to fine-tune metabolic pathways and response to changes in the environment. So isozymes are enzymes that catalyze the same reaction but have different amino acid sequences and structures. Isozymes can have different catalytic properties, substrate specificities, and regulatory mechanisms. This allows cells to fine-tune metabolic pathways and respond to changes in the environment. So in summary, principles of catalysis, enzymes, and enzyme kinetics are critical for understanding how chemical reactions occur in living organisms. Enzymes play a critical role in regulating metabolic pathways, and their activity is regulated by a variety of factors. Understanding the mechanism of enzyme catalysis and the role of isozymes is essential for developing therapies for diseases and designing new biocatalysts for industrial applications. Conformation of protein. Ramachandran plot. Secondary structure, domain, motif, and folds. The conformation of proteins refers to the specific three-dimensional shape that a protein adopts, which is critical for its function. There are several important aspects of protein conformation, including the Ramachandran plot, secondary structure, domains, motifs, and folds. Here is a detailed explanation of each of these topics. So first one is the Ramachandran plot. The Ramachandran plot is a graphical representation. The Ramachandran plot is a graphical representation of the possible backbone dihedral angles phi and psi in a protein. It is used to analyze the conformation of amino acid residues and determine if they are in energetically favorable conformations. The plot is divided into regions that correspond to specific secondary structures such as alpha helices, beta sheets, and turns. Secondary structure. The secondary structure of a protein refers to the irregular arrangement of amino acid residues that form hydrogen bonds within the protein backbone. The two most important types of secondary structure are alpha helices and beta sheets. Alpha helices are formed by a spiral arrangement of amino acid residues, while beta sheets are formed by the alignment of multiple strands of amino acid residues. Then domain regions are there. So protein domains are distinct regions of a protein that fold independently and have specific functions. Domains are often associated with particular functions like DNA binding, enzyme activity, or protein-protein interactions. Proteins can have multiple domains, and the combination of domains within a protein can determine its overall function. Then motifs. Protein motifs are recurring patterns of secondary structure that have specific functions. Motifs can be found in multiple proteins and can be used to predict the functions of a protein. Examples of protein motifs include helix, turn, helix motifs, zinc fingers, and leucine zippers. Folds. Protein folds refer to the overall three-dimensional shape of a protein. Folds are determined by the arrangement of secondary structures and the interactions between amino acid residues. These are several types of protein folds including alpha, beta, all beta, and alpha plus beta folds. The conformation of proteins is critical for their function as they determine how the protein interacts with other molecules in the cell. Understanding the conformation of proteins is essential for designing drugs that target specific proteins, predicting the function of unknown proteins, and developing new proteins with specific functions. Then next topic, conformation of nucleic acids, helix, A, B, Z, tRNA, microRNA in detail. The conformation of nucleic acids, specifically DNA and RNA, is critical for the function as genetic material. Here is a detailed explanation of different types of nucleic acid conformations. DNA helix. The DNA helix is a double-stranded helix that is composed of nucleotide base pairs. There are three main types of DNA helix, A, B, and Z. The B form of DNA is the most common and is the classic Watson and Crick base pairing model. The A form of DNA is a wider and shorter helix, while the Z form of DNA is a left-handed helix that is of a zigzag pattern. Then RNA helix. The RNA helix is a single-stranded helix that is composed of nucleotide base pairs. RNA can form secondary structures such as hairpins, bulges, and internal loops due to complementary base pairing within the same strand. Transfer RNA or tRNA and microRNA, miRNA, are two types of RNA that have unique helical structures. So first, tRNA. tRNA or transfer RNA is a type of RNA that is involved in protein synthesis. It has a distinctive L-shaped structure that is critical for its function. The tRNA has an anticodon loop that reacts with the complementary codon on mRNA during translation, and the acceptor stem that binds to the amino acid that corresponds to the codon. Then miRNA is there. So miRNA or microRNA is a type of small non-coding RNA that plays a role in gene expression regulation. It has a hairpin loop structure that is processed by enzymes to form a mature miRNA molecule. The mature miRNA binds to complementary. The conformation of nucleic acids is important for their function in the cell. The structure of DNA allows for accurate replication and transmission of genetic information. The structure of RNA allows for specific interaction with other molecules in the cell, such as tRNA with amino acids and miRNA with mRNA. Understanding the different conformations of nucleic acids is essential for understanding their function in the cell and for developing new therapies that target nucleic acids. Then stability of proteins and nucleic acids. Proteins and nucleic acids are essential biomolecules in living systems that play critical roles in many biological processes. The stability of these molecules is crucial for their proper functioning in the cell. That topic we will be dealing tomorrow.